This is the lecture on the immune system. The flu epidemic of 1918 killed over 20 million people in 18 months. And right now, we're in the middle of another pandemic. So in this lecture, I want to talk a little bit about our immune system response to the current global pandemic, COVID-19. The COVID-19 pandemic has already killed well over 100,000 people in just 4 months. Why does it act so quickly? Who's at risk? Why don't we have a vaccine yet? Why do most people not get very sick from it? And when people do get sick, what is causing that illness? So what immune system components are being activated that are causing this SARS response? So these are the questions that I want to talk about in this lecture. So first, how does the infection spread? What's the deal with face masks? Should we be wearing them? Should we not be wearing them? What do they actually do? Why do we think we won't get a vaccine for 18 months? What happens in the 20% of people who have a moderate to severe reaction to the virus? And what is flattening the curve? So the first big question I want to focus on is, how does the infection spread? And to answer this, we're going to go over the most basic immune response, the big defense that we have against pathogens. That's our skin. So the vertebrate body is defended from infection by 3 major lines of defense. And I often think of these as sort of a castle siege type game. So we have our walls and moats. That's going to be the skin and mucous membranes. That's our very first line of defense. And that's what we're thinking about when we're thinking about how an infection gets into our body in the first place, are our walls and moats. So can the invaders get in? That's our skin and our mucous membranes, like our eyes and our mouth. We also have roaming patrols. So these would just be guards that are always roaming around the immune system. We think of this as the cellular counterattack. So that's going to be what we're thinking about third in this lecture. So if an infection manages to get past the skin and mucous membranes, we get this cellular-based counterattack. And this is called our innate immune response. And then we also have guards. We have cells in the bloodstream that circulate and look for foreign cells as part of our adaptive immune response, also called the specific immune response. And I'll be talking about that second in today's lecture. So let's look more about how our first layer of response works. This is mostly our skin, but it also includes our mucous membranes. And that's going to be really important in the virus that causes COVID-19, which is the SARS-CoV-2, also called novel coronavirus. Our skin is our first defense against invasion by microbes. Our skin is going to have 2 layers, the outer epidermis, that's the part that's mostly made of dead skin cells that are highly keratinized, that's our waterproof layer, and then our lower dermal layer. Underneath the dermal layer, we have the subcutaneous layer. Cells of the outer epidermis are continuously being worn away and replaced by cells moving up from below. And this is a really great defense against pathogens, because if a pathogen settles on our outer layer, it's going to eventually get sloughed off. This is just looking at our 3 skin layers. We have the dermis of the skin. That's going to be the middle layer here. Let me just get my laser pointer out. So here's our dermal layers right here. That's the middle layer. And this is the layer that has a lot of stuff going on in it. So this is where we keep our things like glands. It's innervated. It's got a lot of capillary beds associated with it. That's right here. Our outer layer, this is the layer that's super, super protective for our skin, for our bodies, but it also doesn't have a lot of activity going on in it. It has this layer of keratinized dead cells that are on the top of it, and these are what are getting continuously worn away and replaced from these layers underneath. And then finally, we have our subcutaneous layer. That's going to be this layer that includes these fat cells. And then our skin also provides some chemical defenses in addition to just this physical defense of being there. We have these oil glands. So here's like a sweat gland, and here's an oil gland over here. These oil glands make the skin surface very acidic, and the sweat glands contain the enzyme lysozyme, which attacks and digests cell walls of many bacteria. So that helps to protect us against bacterial infection. We have these additional surfaces, our mucous surfaces. This includes the eyes as well as our respiratory surfaces, and these are all exposed to the outside. Tears in our eyes contain lysozyme, just like our sweat glands do, to help fight bacterial infections. And then we have linings in our digestive and respiratory tracts. That's what we're looking at here in this picture. But these can be a potential route of entry for microbes. And so we have these sticky mucus traps in them. And these trap most microorganisms in the respiratory tract. And you see the sticky mucus layer right here. So most stuff gets trapped in this. And then in our digestive tract, we have our stomach acids and digestive enzymes that are providing additional protection. We know that the eyes in the respiratory tract are the main entry points for SARS-CoV-2. And so this is looking at the entry of SARS-CoV-2. This is the coronavirus that causes COVID-19. So here you can see that the virus is going to get inhaled into the respiratory tract. This is why they tell you to wear face masks, is to prevent you from coughing out new virus mostly. So the virus gets in through the respiratory tract and then it enters in through these epithelial layers. So it gets past the sticky mucus trap. So here's a zoom in on this. So here is the SARS-CoV-2, and coronaviruses are enveloped RNA viruses. That means they have a plasma membrane studded with proteins as their outermost layer, and they use RNA as their genetic material, not DNA like we use. Envelope proteins, that's on the outside, that blue layer here. Envelope proteins recognize cell surface receptors on our lung tissues. These are the cell surface receptors here. And they trick the lung cells into allowing the viral membrane to fuse with the plasma membrane of the lung cells. The virus then releases its RNA genetic material into the cell. This RNA is translated into viral proteins using normal lung cellular machinery. Since our cells don't have any proteins that can make RNA from an RNA template, the virus carries with it the code for making an RNA polymerase that reads RNA as its template. And so that's what we see here. I think this is this gray one. Okay. This generates more copies of the RNA genome, and so that's here in green. And finally, these RNAs and the proteins are packaged back together, and when they escape the cell, they're going to pinch off a little piece of the host membrane with them, and that's how they're going to be enveloped again. So why are we supposed to be wearing face masks? Are we supposed to be wearing face masks? We catch this and release this through our respiratory surfaces. A big question right now that people are asking is, do face masks help to block transmission? And the answer is we're not 100% sure, but we're wearing them just in case. Coughs and sneezes, we know, can propel the virus past the mask barrier, and that includes past surgical mask barriers as well as past cloth mask barriers. But both of these mask types, as well as the N95 masks, do seem to reduce viral load in the air when the wearer is breathing normally, so not coughing or sneezing. So something that we know is that cloth and surgical masks are probably not very effective in preventing the wearer from catching the virus, but they can help the spread of the virus. So if you're not coughing and sneezing, meaning you're an asymptomatic person, that can the surgical masks and the cloth masks can help you to not spread the virus from your normal breathing. So the next thing I want to talk about is vaccination. So a big question is, why do we think a vaccine won't come for 18 months, right? So that would be a really long time for us to be in lockdown. And to start to get at that, I want to start out with, how do vaccines work at all? And that's going to get into our adaptive immune system. So this is a little meme that's going around. I'm not anti-vax, but I understand why some parents do not want those chemicals in their children's bodies. I think instead of chemical shots, the doctor should give a small piece of the virus so the body can build natural immunity, like the chickenpox playdates we had as kids. And this is a meme going around because this is exactly what a vaccine is. So a vaccine uses a tiny piece of the infectious agent, often called an epitope. It can also use a disabled or a heat-killed virus as a mimic of the infectious agent. And what this does is it activates the adaptive immune response. So when a person gets infected by the real virus, the body's already primed to fight off the infection. And that's what we're going to look at how it works. So just kind of a little background for the COVID-19. You may have heard that we're likely to see waves of COVID-19 until this vaccine is developed against it. And again, the vaccine will activate the adaptive immune response, which I'm going to talk about in just a minute. And this is the special response that our body mounts against specific infections. This isn't a general response like our defense layer in our skin, the mucus in our respiratory surfaces. This is a specific response against a very specific kind of virus, and more specifically against what we call an epitope on a virus. That's a little piece of the virus. It's a shape that one cell in our immune system can recognize. So this is again different from the innate immune response that we're going to talk about later, in that it gets activated by the specific infectious agent. And then this part of the immune system can only recognize that one particular agent in the future. All right, so this is just looking at vaccines in general. So why do we get vaccinated? This is looking at over the years, the different global numbers of deaths for children in each year, and this is by cause of death. So this gray stuff, this is other causes of death. These are not infectious diseases that we have vaccines against. And so you can see that this level has gone down quite a bit, and that's as healthcare has improved in different parts of the world. But the really big thing to notice here is how much smaller these infectious diseases that are treatable by vaccination, how much smaller this proportion has gotten over the years. So over these years, as there's been more vaccination in more countries, and especially mandatory vaccinations in lots of countries, you can see that this share of childhood diseases, deaths due to these childhood diseases, has really shrunken. So this is why it's so important to vaccinate, because we want to keep this number really, really little instead of this really big, huge number that we see here with low numbers of vaccination. And even this number here is with pretty high vaccination levels across the world. So let's look at the adaptive immune response. This is what vaccinations will trigger. So I want to start out looking at the two cell types that are going to be involved in this adaptive immune response. These are T cell lymphocytes and B cell lymphocytes. So lymphocytes in general are white blood cells that are used in the specific or adaptive immune response. The T cell lymphocytes originate in bone marrow. They migrate to the thymus gland for maturation. So we call them T cells because they mature in the thymus, which starts with a T. They recognize microorganisms and viruses by chemical markers on their surfaces, and we call these chemical markers antigens. So this is anything that's not supposed to be in the body that the immune system can recognize. That's an antigen. And when we're thinking about pathogens, these antigens are things that are typically on their surfaces, little bits and pieces of proteins. B cell lymphocytes complete their maturation in the bone marrow, so B for bone marrow. When an antigen is encountered, they produce antibodies. So an antibody is a protein that only binds to one specific antigen. So this is a lock and key model. So the antibody would be the lock, and the antigen would be the key. So they only fit with each other. Antibodies can coat the antigen, and they can also mark a cell that has that antigen for destruction. So an infected cell can't keep producing the virus. Instead, it just gets killed. The first time a pathogen invades the body, here that would be chickenpox, there are only a few B cells or T cells that can recognize those antigens. Binding of the antigen to its receptor on the lymphocyte surface stimulates cell division and produces a clone. And that's what's happening right here. So here we have the amount of antibody increasing, and so we see this big bump here. We call this process clonal selection, and the result is this primary immune response. This is slow to develop. It can happen over the course of about 2 weeks. So that's why the first time, hopefully none of you have gotten chickenpox. I got it twice when I was a kid. It was pretty gross. You get really itchy from it. This was before we had a vaccine. So the first time I got it, I had it or both times I got it, I had it for about a week. I had the itchy sores. And that was the time that it took my immune system to mount this really good response against it. So it took a long time. As a result of this infection, though, there's a big clone of these lymphocytes, B cells and T cells, that can recognize the pathogen, and those are still circulating in your body. So that's what's happening here, is this clonal selection. And so we can have a years-long interval here. Then you get exposed to the same virus again, and now your immune response is really fast. And that's because there are already lots of cells in your body, lots of lymphocytes that recognize this virus and can really quickly respond to it. So this is the secondary response. And in this case, you get antibodies in 3 to 5 days. So this is the really effective response that happens when a particular pathogen is encountered for the second time, or more times than that as well. No invader can escape being recognized by at least a few T cells, because tens of millions of different versions of these T cells are made. Each of them specializes in a different antigen. B cells and T cells interact in complex ways to make sure that only infectious agents that a person is likely to encounter are targeted. Overactive immune systems can lead to allergies and inflammatory diseases. So I'm going to look a little bit at these interactions between the B cells and the T cells. So this is the complicated picture. We're going to break this down in the next few slides. So anything that does not belong in a body can have antigens. Antigen-presenting cells include cells of the innate immune system as well as cells of the adaptive or specific immune system. This is an antigen-presenting cell, and this one's a macrophage, but B cells are also antigen-presenting cells. When this antigen-presenting cell encounters a pathogen, here's this virus that it's encountering, like a coronavirus particle, it's going to engulf it and break it up. And then it's going to put pieces of the virus on receptors on its outside so that other cells of the immune system can see it. So now recall that adaptive immune system cells each respond to just one specific antigen. So most of the leukocytes, the B cells and T cells, are going to ignore this. Very few will be able to actually respond to this. This makes the initial response to a pathogen weaker than the second response. In the secondary response, we have a huge circulating clone of B cells and T cells that all respond to this one particular antigen. Once pieces of the chopped up coronavirus are presented on the antigen presenting cell, they can be seen by the T cells. So here we have the antigen presenting cell. This is our first exposure. And now they can be seen by these T cells here. Cytotoxic and helper T cells can both recognize antigens presented by antigen-presenting cells. B cells are antigen-presenting cells themselves, so they're activated when they encounter a pathogen and present an antigen from it. Then they encounter a helper T cell that recognizes the same pathogen. So to get activated, a B cell needs to encounter a helper T cell that recognizes the same antigen that it recognized. To be activated, a cytotoxic T cell needs to recognize the same antigen that this helper T cell also recognized. This system means that to get full activation, you need two different cells of the adaptive immune system to recognize the exact same pathogen. Once these B cells and cytotoxic and helper T cells have been activated, they undergo mitosis to produce more of themselves that recognize just that one specific antigen. And so the result is this, where we have a bunch of B cells and a bunch of different kinds of T cells that are floating around in your body that all recognize just this one antigen. And we call these memory cells. So we have memory B cells and memory T cells. And that means that when the pathogen is encountered again, the adaptive immune system already has lots of circulating cells that can recognize it and fight against it immediately. I want to focus in for a minute on these antibodies themselves because these turn out to be really important in coronavirus testing. So these plasma cells here, these are the active form of the B cells, excuse me. So the memory B cells can produce plasma cells, and plasma cells will also be produced upon the first encounter with a pathogen. And these secrete different kinds of antibodies. These antibodies will defend against extracellular pathogens by binding to the antigens on the pathogen. So they neutralize the pathogens or target them for destruction. And there are lots of different kinds of antibodies. So here are some of the different kinds. These are the 5 classes of antibodies, and we call these immunoglobulins. So we have these IgMs. This is the main antibody of the primary immune response. We have IgGs. These are the main antibodies in the secondary immune response. Then we have our IgAs. These are secreted into your bodily fluids like mucus, tears, and saliva. We have our IgEs and IgDs, and these are both cell-tagged antibodies. So these stay attached to cells. And so the COVID-19 tests, what they look for is they look for antibodies specific against COVID-19 itself. And so in this test here, we see a negative test. So this person doesn't have IgG or IgMs. So these are IgGs and IgMs. So they don't have antibodies circulating in their body that are parts of the primary and secondary immune responses. This person here had a positive test. That means that they do have these antibodies circulating, means that they were exposed to COVID-19 at some point in the past. Now, this isn't the only type of COVID-19 testing. So this COVID-19 test tests whether your body has mounted a reaction to the virus. There's another kind of testing, and this kind is basically the first kind of testing that we had against COVID-19 before we knew exactly what those antibodies should look like. And this is looking for the presence of viral RNA. So remember, this is an RNA virus. So here we have our virus, and inside this coiled part, this is the RNA in it. And so this is a test that's PCR-based test, and this test looks for whether or not there's any of this viral RNA circulating in your bloodstream. This is just what the PCR looks like. So if these go up really early, that means you have more of it in your blood sample. If they come up late, that means that you have less of it circulating. So just again, looking at this primary versus secondary response, the details of the adaptive immune response explains why we just see a little bit of these antibodies against a virus 2 weeks from the first exposure, but we see lots of antibodies against the virus within 3 or 5 days after the second exposure. The second time, the virus is in our body for too short a time to make us very sick. And in this case, we say that we are immune to the virus. So like I mentioned before, vaccination is a way to elicit this primary immune response without actually getting infected by the virus. In 2000, over 500,000 people died of the measles worldwide. In 2016, the number was down to 90,000, and that was largely due to vaccination. In 2018, this went up again to 350,000 due to fear, as well as lack of access to vaccines. So this shaded area here shows lives that have been saved by the measles vaccine. In some places in the world, there have been major anti-vaccination efforts, and these have led to measles illnesses and deaths. So we saw this in Southern California recently, as well as in other places in our country. And we saw it happen really terribly in Samoa, where last year 83 people died for measles due to low vaccination rates. People are really anxious to produce a vaccine against COVID-19, not just so that the general population can be immune to the disease, but also to protect the most vulnerable populations. These are people who are immunocompromised, especially people who cannot get vaccinated because their immune system does not respond very well. This works via herd immunity. We see herd immunity when there are enough individuals in a population who are immune to a disease that the disease can no longer spread easily. And so that's what we see here in this picture. So let me just get off my laser pointer. So here we see the spread of a disease depending on how many people are vaccinated in a population. So when we have nobody vaccinated, that's sort of our control. When we have about 50% vaccinated people in a population, those are all the people in yellow, you can see that the spread of the disease has slowed down. Oh, and this stopped running for me. When we have 95% people vaccinated against a disease, you can see, it doesn't want to run for me again. You see that the spread is slower, and I'm going to see if I can make this run one more time for you. There we go. Nope, my computer decided not to run it because my computer's feeling really slow today. So why don't we already have a disease? Finding the piece of the virus, that's the concept, that causes the immune system to respond is actually the fast part of vaccine development. In a typical timeline, this part usually takes about 2 years to find a really good piece of the virus, the epitope that causes a good immune response. But clinical trials, that's all of this part here, these clinical trials are where problems are discovered, and this takes up a huge amount of time in vaccine development. In clinical trials, researchers are looking for 2 major things. So one, how well does the vaccine actually work? Does it cause antibodies to be produced against the virus? Does it work in every single person, or is it kind of hit or miss? And then 2, does the vaccine itself have side effects? So let's say there were severe side effects in 1 in 10,000 people who got a vaccine. Well, this seems like a good number. This is a really low chance of you personally actually seeing a severe side effect. But if we look at just the United States alone, if we vaccinate every single person in the United States, this would affect 30,000 people in just the United States. And so a 1 in 10,000 of major side effects is usually unacceptable as far as vaccination goes. And so in that case, if we saw something, if we saw something like 1 in 10,000 major side effects, we'd have to go right back to this preclinical research step and over again. And so it takes a really long time to perform these clinical trials, and then as well as to get licensed by the FDA. And so that's why these vaccines take so long to develop, even if we're working really fast on this first part here. So the adaptive immune response, again, this is the response that the body tailors to a specific pathogen. The innate immune response, that's more general, and you can see this happens much more quickly. So this isn't looking for a specific pathogen, it's looking for any pathogen. Here's our adaptive immune response. This can take days. It's looking for a specific pathogen. And then in hours, though, we get this innate immune response. It's super fast. And that brings me to these two big questions here. So what happens in the 20% of people who have a moderate to severe reaction to the virus that causes COVID-19? And you may have heard that the thing that happens to them is a cytokine storm. So what is a cytokine storm? When an infection occurs, there are a whole bunch of different cellular and chemical defenses that are part of the innate immune response. This is the response that happens almost immediately after the infection occurs. So this includes cells that kill microbes, proteins that kill microbes, and then two things we call the temperature response. That's when you get a fever. And then the inflammatory response. That's where, like say you get a skin infection, your skin will get red and swell up, right? So you may have noticed this, you get like a scratch on your skin, and within, a few hours to a day or so, your skin there gets kind of puffy and red. That's the inflammatory response. In COVID-19, as well as the old SARS, so we call the new COVID-19 is another kind of SARS, so SARS-2. The normal innate immune response seems to go haywire in the respiratory tract, causing an extreme inflammatory response that can lead to lung damage. The innate immune response is a whole set of responses to foreign invaders in the body. And so these are all the different kinds of things it includes, and we call this temperature response pyrogens. This temperature response are these proteins that tell the hypothalamus to raise our body temperature. We already thought a little bit about cells like macrophages. This is a type of antigen-presenting cell, and they are one innate immune system cell type that can kill pathogens. Macrophages kill bacteria by ingesting them. They can also kill viruses. Neutrophils can also ingest pathogens, but neutrophils also secrete chemicals that neutralizes everything living in the infected area, including themselves. So they are basically setting off a little bomb that just kills everything in an infected area. Natural killer cells will attack body cells that are infected. So these natural killer cells find infected body cells and then puncture their cell membranes. And they do this so that the infected cells can't be hosts for pathogen reproduction. So they're basically slowing down the reproduction of the pathogen. So the main thing that we think is causing lung damage in SARS-CoV-2 and SARS infections is the inflammatory response. Infected or injured cells first release these chemical alarm signals, like prostaglandins and histamine. These chemicals cause blood flow to the area to increase and for capillaries to stretch and become leaky. Phagocyte cells migrate to the site of infection and attack the invaders. Many of these cells die, and that forms pus that we sometimes see in infections and wounds. Now, this inflammatory response normally makes these aggressive and cellular chemical counterattacks more effective. And again, they cause the blood vessels to leak fluid and immune system cells into the infected tissue. So here's a picture of the normal inflammatory response, which is a major part of our innate immune system, meaning the same response will happen no matter what kind of pathogen you have in your system. So here we have like tissue injury or a pathogenic microorganism. So we get capillary widening, and that increases blood flow. We get increased permeability of the capillary, that increases the fluid released into tissues. We get attraction of leukocytes, that's our white blood cells. So that gets more leukocytes to the site of injury. And then we also have this systemic response. That's fever and proliferation of leukocytes that happens over your whole body. Now, what can happen in SARS and the COVID-19 disease? So here's SARS-CoV-19 entry into the lungs. So what we see is that we get these T cells activated, and they're going to secrete something called interleukin-6. Interleukin-6 is a cytokine. Cytokines are proteins that are or chemicals that are involved in cell-cell signaling. So when one cell secretes a cytokine, it tells another cell to do something really specific. Interleukin-6 tells quite a few other cells to do specific things, and some of this is this leakiness, the increase in fluid inside of the respiratory tract. So again, this is a cytokine, and it tells the cells around it to mount this inflammatory response that leads to increased fluids. So this can lead to a buildup of fluids in the lungs, and this is when we have the severe stage. So this is not going to happen to everybody. This is mostly going to happen to people who have pre-existing conditions and lung damage already. So when the release of interleukin-6 and other cytokines does extensive damage to the body's own cells, we call this the cytokine storm. And this is an overactivation of the immune response. This is looking at the inflammatory response and how and what happens to lungs in patients who have SARS. So here we see somebody who's got an early infection and they have pretty clear lungs. And now here is at day 15 of the infection. So they've had the infection for about 2 weeks at this point. And you can see that there's a lot of fluid buildup as well as damage in their lungs. They even have some popped blood vessels in their lungs. Here at day 20, you can see extensive damage to the lungs from the fluid buildup as well as popped capillaries. So next up, I want to think about what is flattening the curve. So you've probably heard that the reason we're all staying home is to flatten the curve. Flattening the curve refers to measures that will slow down the spread of infection. One major reason to do this is to not exceed healthcare capacity. Note that most of the models that we have for flattening the curve, at least the early models, don't account for healthcare workers themselves getting sick. They're mostly looking at numbers of ICU beds rather than looking at the numbers of people who can actually operate healthcare in those ICU beds. Distancing measures will reduce the rate of healthcare workers getting sick as well. So here we have our number of cases, and here this dotted line is the capacity of a healthcare system. So this would be normal infection, and you can see that most of the models for COVID-19 show that we're going to exceed the healthcare system capacity without protective measures. With protective measures like social distancing, we are trying to flatten this curve. So this is reducing the spread of infection early on. So even though the area under this curve might be the same, meaning that eventually the same number of people will get infected, with flattening the curve, we're reducing the speed of this. And so that people's infections are getting kind of more spread out over time so that we don't max out the healthcare system capacity. So this little video, hopefully, here it illustrates projections of infection rates over time with different types of interventions. See. Oh. No. Now, this was definitely working when I tried this out, but I think I'm running too many programs right now. But we don't totally need the illustration for this. I can just, the video for this, I can just show you the illustration part. So this is looking at different types of intervention. So in here, the orange are infected people, blue are uninfected people, and pink are the people who are recovered, who are presumably now immune to COVID-19. So here's the free-for-all. Here's an attempted quarantine, and in this case, people got sick of the quarantine, so they went back outside. So you can see the curve is flattened at the beginning, and then we have another spike of infections. This is under moderate social distancing, so that's the kind of lockdown that we're on now in California. So we're under moderate distancing, where a lot of people are staying at home, but not everyone. We're still going out for groceries. We're still allowed to go out for walks. And you can see that the curve is flattened quite a bit in this. And here under extensive distancing, this would be a hard lockdown. Maybe you are not allowed to leave your house. You get groceries delivered to your house. You don't go out, almost no one goes out for work. And so you can see that this really, really flattens the curve. So again, why flatten the curve? Besides sparing their healthcare system, which is huge, this buys time for healthcare workers to procure protective equipment as well as life-saving equipment. Flattening the curve also gives scientists time to develop new treatments to fight the symptoms of COVID-19, as well as to develop a vaccine. Existing drugs like anti-inflammatories, as well as drugs that break down mucus in the lungs, so that's what we're looking at here. We have this hyaluronidase that's going to break down stuff in the lungs. We need to test all of these. They're already being tested on patients that are suffering from severe COVID-19 SARS. But we don't know really what works, what the side effects are for a lot of these in people who have COVID-19. So these are all drugs that that people have used in other kinds of diseases. So we know what the general side effects are, but there can be slightly different side effects for people who have a different disease. So again, flattening the curve is buying us time to test these out, to see what works, what doesn't work on just a few people before we kind of roll it out to a lot of people getting sick all at once. So this figure is way too complicated to go into in detail, but it shows a lot of the different existing interventions. These are all things that are already FDA approved that could potentially be used to treat the severe COVID-19 cases. The more we know about how the novel coronavirus affects our bodies, the more of these different interventions are possible. I just want to briefly mention some other things that can happen when different components of our immune system become overactive in different tissues. An autoimmune disease is when the body attacks its own tissues. For example, multiple sclerosis, type 1 diabetes, rheumatoid arthritis, lupus, and Graves' disease. One of the most common kinds of autoimmune diseases are allergies. So in allergies, our body mounts a major defense against harmless antigens. So here this allergen is getting detected by a B cell, and the B cell is mounting a response to it. The allergen is also going to be noticed by a mast cell, which is a part of the innate immune system. And this mast cell is releasing histamines and other chemicals. This gives us an allergy. So when there are B cells around that recognize particular allergens, those B cells can tell these mast cells to start to release histamine and other chemicals, and this gives us all of these bad things that we associate with allergies. So for example, asthma, this is a form of an allergic response in which histamines cause the narrowing of air passages to our lungs, so it's hard for us to breathe. So the inquiry and analysis for this lecture is immunity antigen specific. So here we're seeing the levels of anti-A or anti-B antibodies. So the red is the anti-A antibodies, and the blue are the anti-B antibodies. So this primary response is going to be elicited by the injection of antigen A, and the secondary response here is going to occur with reinjection of antigen A, and at the same time, the scientists are injecting antigen B. So what we want to know is, one, the speed of the primary and secondary responses to antigen A, which reaches maximal antibody response quicker. And then two, does prior exposure to antigen A have any effect on the speed or magnitude of the primary response to antigen B?